We are picking up with where we left off at metaphase 1 here. So in metaphase 1, we talked about how the tetrads form, and this is called synapsis, where the tetrads form along the middle of the cell. And we talked about how crossing over can happen, where the homologous chromatids swap segments of DNA, which increases variation. So now we are going to look at the next phase, which is anaphase 1. And I'm going to switch over to black here. And just like in mitosis and anaphase 1, the chromatids are going to be pulled apart from each other by the spindles. And so this is another really interesting phase because something else happens that can increase variation. So we'll draw our centrioles again and our spindles. So in anaphase 1, the chromatids are pulled apart, but they're not always pulled apart the same. So it's completely random. And there's a couple of phrases here that address this. The process of pulling chromosomes apart is called disjunction. Pulling chromosomes apart. And so, excuse me, in this case, we'll show the 2 red chromosomes get pulled to the left, and the 2 green chromosomes will get pulled to the right on chromosome 1. But remember, we had our crossing over occur, so we'll add the crossing over segments here. On chromosome 2, we will say it is the opposite, or we'll say that they got mixed up, and 1 red one got pulled to the left, 1 red one got pulled to the right. 1 green one got pulled to the left, and 1 green one got pulled to the right. And in number 3, we're going to demonstrate a process that happens rarely. I think that maybe 1 in 700 times this would happen. And this is called non-disjunction. And in non-disjunction, one of the chromosomes gets stuck. So we wind up with only 1 chromosome going to one side, and one chromatid, and the 3 chromatids go to the other side. So this process right here is called non-disjunction. And this is what results in genetic conditions where a human has an extra chromosome, such as Down syndrome, where there's 3 copies of chromosome 21. So this is called non-disjunction. And the chromatids don't completely separate. So again, that's rare, but we're demonstrating it here so that you can see how it plays out. So notice that each of these chromosomes, chromosome 1, chromosome 2, and chromosome 3, separated independently of each other. And what the result of this is that it causes huge variation in offspring because every single time meiosis occurs, or each of the 4 cells produced by meiosis is going to be different than the other 4 cells produced by meiosis. And so the name for this is independent assortment. Okay, in an independent assortment, each chromosome should erase that. Is separated differently and randomly. And this idea of independent assortment increases variation. Okay, so that's anaphase 1. Telophase 1, and really the rest of meiosis is really straightforward. Nothing else interesting happens. So in telophase 1, in cytokinesis, these two cells, or the chromosome, sorry, the cell is going to pinch off into two cells. Our nucleus will reform. And then inside, I'm going to just take a peek at how we sorted these out. And then inside, you've got chromosome 1, chromosome 3, chromosome 2, and chromosome 1 has a little bit of crossing over. Chromosome 2 has a copy from each parent, and chromosome 3, remember, has a little bit of the green, a little bit of the red there. And then chromosome 1, on the other cell, we have the crossing over. Chromosome 2, and then chromosome 3, we just have the one copy of the green one. That one shouldn't be any red there. Okay. And so this will now continue to meiosis 2. And we are just going to gloss over this. So meiosis 2 goes metaphase all the way through telophase, just like what happens in mitosis, but with two cells. But it's going to result in a critical difference at the end. So let's take a look at how this happens. So we are going to start out with the result of telophase. Draw our nucleus again. And we will draw our telophase cells again. And so what's going to happen here is that each of these cells is going to go through the four phases: metaphase, or prophase, metaphase, anaphase, and telophase. So each will be dividing, we'll make that black, into two cells. But each of these cells is going to have the chromosomes split. So this chromosome 1 on the left-hand side, one of those chromatids will go to the left cell, one of those chromatids will go to the right cell. And again, with independent assortment, it doesn't matter which way they go. So I'm going to draw this one going here, this one going here with a little green tip on it. Chromosome 2 will also split. So I'm just going to draw the red one here and the green one here. It doesn't matter, you can switch it. And chromosome 3 will also split, but remember, chromosome 3 had non-destruction occur, so one of these chromosomes is going to actually have two chromatids. And then the same will occur on the other side. This cell on the right-hand side, the original cell, will split in half, and we will have the red go to the right this time, and the green to the left for chromosome 1. put that little piece of crossing over on there. And then chromosome 2 will have it split the same. We'll have green go here, red go over here. And then chromosome 3, there's only the one, so we'll just have the green go to this side, doesn't matter. So what I want to point out that's critical is that each of these cells now has half the original amount of DNA. So each of these cells now is a 1N cell. So we now have 4 1N cells. And they are all different from each other. And so these cells are going to go on to become sperm or egg. And when they fertilize, this is going to increase variation because you have no idea which one of these. So this explains why siblings will all look different from each other unless they are identical twins. So that about wraps up meiosis.